In this webinar, I'll talk about two innovative novel products related to endoplasmic reticulum, ER-seeing and ER protein capture kit. ER protein capture kit enables the isolation of ER-associated proteins, not ER itself. This webinar has two parts. Part 1 is ER-seeing section, which we will discuss what is ER and ER-seeing. Part 2 is ER protein capture kit section, which we will discuss about conventional ER protein detection methods and ER protein capture kit. First of all, I will begin to talk about what is ER in Part 1. What is endoplasmic reticulum, or ER? ER is the largest organelle and has the most complex structure in cells. There are two types of ER. One is rough ER and the other is smooth ER. The rough ER has rough appearance because it has lots of ribosomes on the surface of ER membrane. On the other hand, smooth ER does not have ribosomes and has more tubular structure. The main ER functions plays essential roles in protein synthesis, post-translational modification, origin of protein secretory pathway, lipid and carbohydrates metabolism. Subsequently, I'll talk about one of our new reagent, ER-seeing. Our ER-seeing and ER protein capture kit are based on the ER-localizable reactive molecule, or ERM technology. ERM is a small compound which has two units, a rhodotype green fluorescent dye and a thioester type protein labeling group. The green fluorescent dye has a high affinity to ER membranes. Immediately after adding this reagent to cells, will selectively and quickly accumulates into the ER. Once accumulated in ER, the labeling group of ERM randomly reacts with amino acids and ER proteins and forms covalent bonds with the fluorescent dyes. In other words, the rapid migration speed of ERM to the ER, there is zero time for protein labeling. As the ERM accumulates and concentrates inside the ER, the proteins become conjugated with the fluorescent dye. This is the reason why ERM technology labels ER-associated proteins selectively. The reagent's name of ERM is called ER-seeing and useful for live imaging of ER protein movement. The most well-known conventional method for ER staining dye is glibenclamide-based dye. Glibenclamide is a specific inhibitor of ATP-sensitive potassium channels and FDA-approved drug for diabetes. Because ATP-sensitive potassium channel receptors are numerous on the ER, glibenclamide-based ER dyes can visualize ER structures. However, pharmacological activity of glibenclamide negatively affects potassium channel function and change potential balance in the ER. This page shows comparison between glibenclamide-based dye and ER-seeing. Though glibenclamide-based dye can visualize ER structures, glibenclamide negatively affects ER function. In addition, the dye reversibly binds to the receptor and may detach from the receptor gradually at watch steps or medium change. This suggests that glibenclamide-based dye is not suitable for long-term imaging experiments. On the other hand, ER-seeing has high ER selectivity, high retentivity, and little toxicity. ER-seeing labels ER proteins which are located on ER membranes and ER interior. ER-seeing exhibits little pharmacological effect on ER functions, and fluorescence can be observed even after medium change. Here are staining photos using ER-seeing and glibenclamide-based dye. HeLa cells were treated with both ER-seeing and glibenclamide-based dye, and as a result, the stain was highly overlapped. However, the result after medium change was totally different. No signal was observed from glibenclamide-based dye after medium change. However, high signals were observed by ER-seeing. Thus, ER-seeing is a powerful tool to monitor ER structures in live cells with little pharmacological effects. Here is a summary of ER-seeing. ER-seeing has a high specificity for endoplasmic reticulum. Compared to glibenclamide-based dye, ER-seeing has lower pharmacological effect than ER-seeing and can observe the behavior of the normal physiological ER conditions. In addition, ER-seeing forms a covalent bond to ER proteins and suitable for long-term imaging. Here's the product information. catalog code FDV-0037 and sufficient for 8 plates. Let's move on and discuss Part 2, ER Protein Capture Kit section. Please note that this kit allows capturing ER proteins which are located around ER but does not isolate ER itself. First of all, I'll talk about conventional purification methods of ER proteins. The ER plays important roles, but isolation methods are very limited due to this complex structure. Cell fractionation by ultracentrifuge is one of the classic approaches to isolate ER. In this method, ER fraction can be concentrated mainly in the P3 fraction. However, P3 fraction contains not only ER fragments, but also other fragments such as endosome and Golgi apparatus. Additionally, ER structure is ruptured during homogenization and part of ER contents will escape to the outside of the ER. Therefore, the classical cell fractionation method is insufficient to purify ER selectively. To this date, only a few isolation kits are commercially available on the market. Please note, these kits aim at isolating ER itself to obtain ER proteins. One kit allows to obtain P3 fraction by centrifugal separation. As mentioned earlier, this method is not able to obtain the whole ER selectively. Another kit allows to concentrate and isolate ER with spin columns. However, this kit is compatible with liver tissue only. On the other hand, our ER protein capture kit aims at isolating ER proteins directly. I'll explain this in details later, but ER protein capture kit allows the direct isolation of ER proteins selectively by immunoprecipitation. Let's talk about ER protein capture kit. Our protein capture kit is based on the ER-seeing as discussed earlier. Let's look back earlier about ER-seeing to refresh on the labeling mechanism. Because the green fluorescence dye has a high affinity to ER membranes, immediately after adding the reagent to cells, it quickly accumulates into ER. Once it accumulates in the ER, the labeling group of ER-seeing reacts with amino acids and ER proteins randomly and forms covalent bond. That's why this technology enables to label ER protein selectively. This data shows the specificity of ER-seeing. HeLa cells were treated with ER-seeing and organelle markers for ER, lysosome, mitochondria, and Golgi. ER-seeing is highly overlapped with glibenclamide-based dye, but does not correlate with the lysosome and mitochondria markers. On the other hand, ER-seeing is slightly overlapped with the Golgi staining. So why does ER-seeing slightly overlap with the Golgi marker? There's an explanation why ER-seeing is slightly localized in Golgi apparatus using this picture. As mentioned in the earlier slides, ER plays an important role in protein synthesis, especially secreted proteins. So plasma membrane and extracellular proteins are first synthesized in ER and subsequently transferred to the plasma membrane or extracellular space via secreted pathway. This picture shows secretory pathway from ER to Golgi and exosomal vesicles. Some of the ER-seeing stained proteins in ER may migrate into Golgi apparatus, and these proteins can be slightly detected, which this is a very reasonable process. Actually, when cells were pre-treated with ER-Golgi trafficking inhibitor, Golgi staining by ER-seeing was dramatically decreased. Please note, ER contains secreted proteins, including membrane proteins and extracellular proteins. So ER-seeing identifies ER-resident proteins primarily, but as labeled proteins migrate to Golgi apparatus via secreted pathway, there will be minor Golgi staining, which can be detected. Returning back to how we can isolate ER proteins by using our kit. As I explained earlier, ER-seeing conjugates onto ER proteins tagged with green fluorescent rhodo dye. So if an antibody specific for the green fluorescent rhodo dye is developed, rhodo-tagged proteins can be purified by using immunoprecipitation. And we have developed an anti-rhodo antibody to purify rhodo-tagged ER proteins using immunoprecipitation technique. Here you can see the overview of our ER protein capture kit. In this kit, there are two components, ERM and anti-roto green fluorescence antibody. The basic principle of how ER protein capture kit can purify ER protein is, once ERM is added to live cells, it accumulates into the ER and labels green fluorescence dye to ER resident protein selectively. After preparing the cell lysate, green fluorescence dye tagged proteins by ERM are selectively purified by immunoprecipitation with antibody. Purified proteins are separated by SCS page and by LC mass spec based proteomics or Western blot method. This kit allows to purify both ER resident proteins and ER associated proteins, including secreted pathway proteins, with easy procedures and no special equipment. Here is experimental data which shows comprehensive identification of ER proteins by LC mass spec proteomics in HeLa cells. ERM, an anti-roto antibody, was used with HeLa cells, and as a result, a total of 146 proteins were identified in this experiment. Among 146 proteins, 63 proteins were known as ER-resident proteins, and 73 proteins were categorized in the secretory pathway, such as membrane, Golgi apparatus, and extracellular proteins. As ER is the origin of secretory pathway, proteins in this pathway can also detect around 93%. Furthermore, the original paper revealed some remaining proteins are actually novel ER proteins. Another experiment example is ER stress quantitative profiling. ER stress is one of the interesting topics in cell biology and indicates abnormal function of ER caused by extracellular drug stimulation. ER stress is also seriously related to various diseases such as ALS, Alzheimer's diseases, and so on. Here is a demonstration under a chemical-induced ER stress. I will skip talking about the experimental details, but more importantly, these results show some protein increase by ER stress and other proteins decreased by ER stress. This experiment and using this kit allows us to evaluate the change of ER localization of protein amounts under extracellular stimulation. So in summary, ER protein capture kit can isolate ER proteins with high selectivity. No need for special instruments or techniques like ultracentrifuge or density gradient technique. Just perform immunoprecipitation to purify ER proteins. The procedure can be completed in 1 day. And here is the product information, catalog code FDV-0038, which is sufficient for 20 assays. Thanks for your time listening to this presentation. I hope you find this overview of our innovative reagents and kits useful in your research. If you have any questions, please feel free to contact us at any time. And for additional information such as downloading manuals, availability, and pricing, please communicate with your regional contact. For Japan, you may contact Funakoshi Co. For United States and Canada, you may contact Diagnosing. And for all other countries, please contact your local Funakoshi Co. distributor.